awesome guy, right? Ability to part the veil with faith. Faith is a very important principle of the gospel that frankly we could study our entire lives and never get to the bottom of, but yet we teach it with 1 or 2 sentences sometimes. And so mastering it, when you know you've mastered it, go ahead, be like Brother Jared, part the veil. Because once you have, there shouldn't be anything withheld from you. Sometimes we think on the other side, I'm going to get to the other side and I'm going to remember it all from the premortal realm. But why can't that be correct?
Well, we're also going to be in a state of probation until the final judgment.
We are, and we know that the people that are there that are in what we term spirit prison, right? The Savior went to organize so that they could be taught the gospel. Well, they wouldn't need to be taught the gospel if the veil was lifted when we died, right? It isn't. So, how are you going to remember everything? When you're there, when you have faith enough to do it, right? So might as well do that now. Good plan. I'm working on it. We'll see if I get there. Yep. All right. Membranes. Parting the veil. Close enough, right? All right. Monomeric lipids. Yeah. They really don't like to go into water. Virtually insoluble in water altogether with these type of shapes. Even though they have some polar pieces to them, right? So, a micelle. Everybody good with the micelle structure? It was formed using what effect that we talked about at the very beginning?
Hydrophobic.
Hydrophobic effect, and all that entails in review, right? So there is that micelle, a structure that has the head group that's larger than the tail of the ischial chain. A few thousand of those inside a micelle. Then we get a lipid bilayer. Now that's what we're going to start talking about, that phospholipid bilayer that's our membrane. So it's slightly different, okay, but largely composed of the same thing. Notice this one has 2 acyl chain groups sticking down off the end of it. Does that make sense based on what you just studied about glycerol phospholipids? Yeah, that's a perfect match of what we expect to see. Are there mono ones? Yes, but most of them are looking just like that. So this is what's called a vesicle. A vesicle is different from a micelle in the fact that it has interior aqueous domain. That has a vague resemblance of what? A cell, right? And any of the organelles inside the cell. So when these form, there are 3 general ways that this happens. We can have a bilayer that actually turns around and folds back on itself to make this aqueous cavity in the middle of our vesicle. We could actually have cell division that's occurring. And if I have the micelle volume that's too great, now this is what's weird, but what would happen if I kept trying to add fatty acids here to my micelle? Eventually the tails can't reach to the center, so it's going to flip on the other side, and we actually create that surface. That's also a viable way that they're formed. So there are the 3 ways that happens. There is a limitation to the size that can happen. We can't have negative space that we create with them. So that's interesting. You also begin to see over here in D, and sorry guys in the back, but I'm going to show you bigger ones in a second. these shapes. There are different shapes of fatty acids, and you may not have considered that previously. Here's a little bit of knowledge set. This is E. coli. There's the membrane. You're actually looking at it under a microscope. This is about 4 nanometers across, 40 angstroms, right? So that's a pretty interesting view. When you look at those membranes itself, they have a function, right? To keep nutrients in and to keep garbage out, right? Such as waste and toxins. We also use them, we create gradients, right? Electrochemical gradients inside of those, proton gradients. And we use those to actually create nerve stimulus, to create things like our ATP using the proton gradient pump, right? So these are all part of what we're looking at. There's a more broad definition of a membrane, 30 to 100 angstroms. We will get to talking about very soon here lipid rafts too, which are thicker. Gosh, what's the charge inside of a cell? Negative. Okay, well, that's interesting. Okay, so polarized. And there's a right side and a wrong side out in it. So let's take a look at these major components of plasma membranes. Now what's interesting here, as you look at it, you've got protein amounts varying, phospholipids varying, sterols varying. Why aren't all membranes the same?
Different functions.
Isn't it to do with temperature?
Okay, we're going to look at temperature in just a second, but here we're talking about different function.
Oh, okay.
Different function, right? And that's really what we get interested in. With the different composition of just lipids, let's isolate on this red one on purpose because you're going to run into it in just a second on your worksheet. It's called cardiolipin. Now, we looked at cardiolipin before. This is the first time I'm going to show you an entire cardiolipin all drawn. There's the glycerol in the middle, which is connected to 2 phosphates. And then those phosphates are onto a glycerol and another glycerol and 2 acyl chains. 4 acyl chains in all. That's the cardiolipin. Used in membrane structures for stability because they're monstrosity. Look at the tissues that they're involved in. Where are those cardiolipins really abundant in?
Mitochondria.
Mitochondria. What's going on in a mitochondria that I'd want to have a massive structure like that? Electron transport, yes. But is it the electrons that I'm worried about?
The pumps, though.
It's the protons.
Yeah.
Yeah. That's what we're thinking of, controlling those protons. Again, you can see now different shapes of different fatty acids, different lipid structures. And sorry, they're abbreviating a whole bunch of them, right? That cardiolipin I just showed you, this is actually what it really looks like.
Whoa, what?
It's probably not what you were imagining. And no, they're lining this membrane where I've got protons that I'm moving in and out of my cardiolipin. They're a bicyclic structure with those 4 acyl chains coming in there, okay?
Oh, that makes a lot more sense, okay.
All right, so some cool things to sort of get and start envisioning. Conical shapes, I can have the curvature of a membrane forced one way or another depending on the shape of my fatty acid. That's interesting. There is different things on each side. The one thing I would draw your attention to that's really quite interesting in my world is this one called phosphatidylserine. Now, the reason I bring up phosphatidylserine is because if it gets on the wrong side of the leaflet of the membrane, it signals something. Is anyone familiar with what phospholipid, yeah, what do you think?
Apoptosis or in phagocytosis? Or like being
It-
engulfed by a macrophage, sorry.
Yeah, but it really is signal for apoptosis. I've lost control of my cell, and the brakes are off. I'm not in control anymore.
I need to
So
die.
if it puts that on the outside, it says, kill me. Whatever way we're going, it's like, I'm on death row here, get rid of me. Save the rest of your cells.
So is that something that happens on purpose? The cell will put it on the outside?
On purpose, and then sometimes accidental.
Okay.
Either way, if it's left there, it creates a problem.
Okay.
Notice it's also on the outside of this inner vesicle.
So if it was on the inside of the inner vesicle, it
It
ends
would
up on
be a
the
problem.
outside. Yeah, okay.
Okay, so where is it located? This is talking about each of those compositions again in different cell types, so you can get that idea. There's that phosphatidylserine again, also activates blood clotting, marks for destruction. Wow. Okay, was that in green, by the way?
Yes.
Wonder why I do something like that. Okay, functions of proteins, receptors, channels, enzymes, good. This is where I start seeing things that sometimes create a little confusion but are not meant to be. I've got a peripheral protein, amyotrophic protein, and an integral membrane protein. The integral membrane protein is probably the easiest one to understand. It's a protein that's embedded itself in the membrane such, with its hydrophobic domain so embedded, that you can't get it out without adding a detergent. Soap, which is essentially substituting for the membrane itself. So that's the only way to get those babies out. Often when we look at structures of these, like the one that my major professor had purified, the structure and its crystal structure are also with detergent molecules surrounding it.
Wow.
And just about 7-8 years ago, they started to figure out how to do them in NMR by making a fake nanodisc where they band it with proteins, but they'd have this little nanodisc, and then they could spin the nanodisc, which they made synthetically, and then they could do NMR on it.
Okay, cool.
That's interesting. But these two right here are really confusing. Amyotrophic and peripheral proteins. The peripheral proteins, I can knock those off by doing what, according to this? Change the pH, add a chelating agent, urea, carbonate. I'm disrupting ionic interactions on the surface, and they're falling off. So they're not hard to get off, right? In fact, by accident, you might get them off. Okay? But these amyotrophic proteins are different. They have something that isn't shown here very well, but is shown over here much better, which is still an amyotrophic protein. These little red dots here, those squiggle lines are a lipid anchor. Okay? And lipid anchors are embedded in the membrane itself. They're hydrophobic fatty acid material, and they're covalently attached to the protein material, such as my glycophosphatidyl's and inositol. Yes?
So if I were to put this in order of being able to separate them, we'd go peripheral is easiest, amphoteric is middle, and then integral is the hardest.
Right. In this one, the way that we get them out, what do you think?
Enzymes probably.
Enzymes. That's right. Enzymes specifically designed to cut the interface that we're looking at. Okay? There's about 6,718 membrane proteins known in humans.
Dang.
Of the 21,000 we know. There's a lot that like the membrane. Okay. There you're looking at peripheral proteins, knocking them off. There you're looking at the epitrophic, and yeah, lipid anchors, knocking those off. Let's zoom in quickly to lipid anchors. Here I'm looking at them. Wow, a palmitoyl group, which is a C16 fatty acid, linked in this case to a cysteine or serine. Virtually the same linkage, just the sulfur versus an oxygen, and I'm making a covalent link there with the fatty acid. Essentially a thioester or a ester. Okay? I've also got myristoyl groups attached to at the amine. Okay, that can also happen. I've also got something known as a farnesyl or a geranyl, and it's got this funny shape with dashes off the side. Does anyone remember? We talked about it just last week. What's this geranyl and farnesyl from? What was it found around? It's a precursor to creating one of the molecules that you're supposed to know about the class of them.
Isoprenes is what we're talking
So it's
about.
made out of isoprenes, okay, isoprenoid units. Isoprenoid units were used to make what structure?
A bunch of them.
A lot of them really ended up made, you're correct.
Sterols, though?
Sterols was the overarching end goal, though there were several that turned into fatty vitamins and other materials. Geranyl and farnesyl are on the way to making a sterol, and they're used as lipid anchors. So all of these are lipid anchors. That's what you're looking for. Okay?
Okay.
Amphitrophic proteins have those. Okay? Farnesylation, promising therapy. I mentioned the other day too. RAS and Rho actually have on them these geranyl and farnesyl. Okay, and if you don't actually use the farnesyl transferase to attach it, or if you block it because it doesn't have them, because I block statin drugs, which stop the synthesis of cholesterol, but because it's an earlier precursor even than farnesyl and geranyl, I'm stopping farnesyl and geranyl from being created in high abundance. By the way, statin drugs lower your actual cholesterol by about 30%. So you're not talking about knocking anything out. You're just slowing it down. And that helps slow down the progression of aggressive cancers in some instances. Okay? All right, integral membrane proteins, I mentioned detergents. What is a detergent made of? Tell somebody else. Let's wake up for 2 seconds, and then we'll go 5 more minutes, and we'll turn you loose.
It forms micelles that have our carboxylate group like so long.
Yep.
And it's basically
Grabs it.
except for it's not vegetables, right? What's specific? The protein
Wow.
That's good. Everything.
Holy cannolis.
Detergent's basically just lipids, but like with different head groups.
Yeah, there you go.
But it's a different polar head though. 'Cause
Okay.
it's not a glycerol.
Hopefully you're not floundering too bad. Again, a laundry detergent falls in this. Your dish soap falls in it. It's something that has a very polar head group, usually a sulfate or a phosphate, okay? Which is very soluble, loves water, and then I've got a hydrophobic domain. There's some acyl chain, so SDS, sodium dodecyl sulfate, yes. The sulfate's the polar head group. Sodium's offsetting the charge, it just falls off once I put it in solution, and some acyl chain. Dodecyl, right? You can guess that it's how long? 12.
Yeah.
And you'd be right. Okay.
So I want to confirm I'm understanding this properly. Main difference then between like a detergent and a phospholipid itself is the lack of a glycerol connecting the two groups? Okay. Otherwise, so that's why it's really good at getting it off.
Yeah, and we did talk about some natural detergents that are found inside your body made from sterols. You guys remember what those were? Bile salts, okay?
Yeah.
Made of a sterol connected to an amino acid-like structure, which forms naturally what we're looking for. Okay. Integral membrane proteins embedded. If I have 7 domains inside of them, they come up with something known as a GPCR. That's actually how all of them are classified, that are GPCRs. But there are different types of integral membrane proteins depending on how many times they cross that membrane. It's interesting, but this is made out of a protein structure. What do you think's on the outside of that? Alpha helical structure. R groups that are non-polar. But thank you for clarifying, because earlier on, that would be a really easy trick question that might come back on a final. Where do R groups reside?
On the outside.
On the outside. Okay, that makes perfect sense to us, right?
So we're
Okay.
talking about alanines, glycines, those kinds of guys, right?
Yeah, so, and tryptophans, right?
Tryptophans, yeah.
You're
Cool.
right, okay. Looking here, red, tyrosines right here, and tryptophans, where are they actually located? Are they right in the middle, or are they? Yeah, they're actually at the edge, and that's because they have a hydrophobic and hydrophilic domain. Hard to put that tyrosine's hydroxyl embedded inside the membrane. Even though the phenyl group has no problem, the hydroxyl doesn't like it. So that's where they tend to reside.
Not bad.
Yeah.
Kind of more an application question, but like, we see, again, that idea that the non-polar stuff more towards the inside with the hydrophobic effect, when it builds the cell membrane and like these proton pump channels go in, does that affect like their folding? Like, so like it wouldn't, normally that sequence of amino acids wouldn't normally fold that way, but because it's in a membrane, it's gonna get into that change where it can perform its function that way.
Yeah, so how would I have to protect it until I could get it there?
Chaperonin.
Add a chaperone. Now let me get you even the higher level. I mentioned that really 7, 8 years ago, we came up with a new technique where we actually did NMR structures of these. And prior to that, we had X-ray crystal structures where we had a crystal, but we had detergents embedded with them. It was when we started making membrane proteins in their membrane domain and looking at them as an NMR in a natural pH environment instead of a crystal, that we realized that some of the structures that we had protein structures for in the database that still reside there, the active group, the site where where either the ligand would bind or the substrate would bind, was wrong. It changed its shape when it resided in a true membrane. In fact, some of them, we were floored. The actual reaction, the molecule has to get into the membrane to react in order for that to happen, which blew our minds. Why would I say that? Because this next unit that we're going to go through will also be dealing with receptors and signaling pathways. And even though the book's not going to go into that detail for you, realize that if you're talking about creating a drug, or you're talking about a synthetic path, a natural pathway where you got a signaling molecule, it's really important to try to mimic it to understand what is the actual protein I'm looking at. Where is the active? We talk about that misnomer of the lock and key, but it's still, you've got to understand the shape that you're trying to bind that will then morph itself as it does bind. So, yeah, good question. Hopefully I helped.
No, yes,
even though
yeah.
I went over and above, right? So you'll look at some of these, which are looking again at those integral membrane proteins. These are something called a hydropathy plot. Now these show up in actual papers. People take the primary sequence of a protein, which is what?
Just amino acid sequence.
Just the amino acid linear sequence.
Yeah.
And they find regions of it that have a whole bunch of amino acids that are hydrophobic in a row. And if it's a long enough strand, they start to say, I bet you that's actually spanning across the membrane. Now, these are real structures. Both of these over here are real structures that you're looking at, and there's the domain. Here you see 1, 2, 3, 4, 5, 6, 7 domains, a GPCR of a rhodopsin. Oh, okay. There you're looking at a glycoporin molecule. Hydropathy plots are used using the hydropathy index. Ironically, you saw this when you memorized your R groups and your pKa's. But we ignored this column, which had hydropathy indexed. And what do you look at? You go, oh, serine. Oh, threonine. Oh, I'm looking at asparagine, glutamine. These ones would be what? Polar or non-polar?
Polar.
And they're negative. And when I'm looking at values that are positive, oh, I'm looking at a valine, I'm looking at leucine, isoleucine. Those ones must be non-polar. That's the values that are then positive, they're non-polar, polar. That's how they create them. Make sense? And then residue number, that's a fancy word for
Amino acids.
the amino acid in sequence. First amino acid, second, third, up to 50, up to 100. Yep, that's all they're doing. What amino acid was it if I were going from the N terminus to C terminus? Okay, cool.
So on that chart, the one this time around, what's more important to remember on this chart is the hydropathy index.
That's right. So I just grabbed the same slide, but that's actually what they're referring to create those hydropathy plots. Okay.
We don't need to memorize
You
the
do not
values.
need to memorize them, but you should already have learned your amino acids in such a way that if I say is it hydrophobic or hydrophilic, you'd easily, not numerically rank it, but know it is polar or it isn't, right?
Okay.
That should already be in your working memory, okay? Remember things like right-handed helix, 3.6 residues, 5.4 angstroms per turn. Gosh, I wonder if I were counting the residues, if I could actually figure out how far across the membrane would be. I bet you'd have to do that in just a second. Okay? Yep, and that math's up there. Awesome. Anti-parallel, could you estimate a beta barrel crossing the membrane? I bet you could. That's cool. And then we'll get to the fluid mosaic model right here. This is, I gotta let you guys go. You go forward right here. There is other things right here, membrane phase changes, and then also you'll start here with the temperature of fatty acids. This is just E. coli at different temperatures and what it's doing to its fatty acid composition. Why don't you start working on that worksheet? Get as much of those notes filled out as you can, and just a good way to review some of the things we've covered today, get those locked in, recognize these are the important things.
You got it.